Bio 107. I am going to finish up the lecture that we started today in class, talking about the modifications and additions to Mendel's principles. So some of you came up to me after class and asked about, well, what about crossing over and things like that? Mendel's principles don't really account for that. And the answer to that question and or statement is, yeah, good point. Mendel didn't really know about the genetics. Genetics wasn't a thing when he was doing his stuff. He was using very simple organisms and asking very simple questions. But we all know that genetics is not nearly as simple as a 1 to 2 to 1 ratio necessarily, because what about crossing over and other things? So the things, the different additions that I'm going to talk about are incomplete dominance, codominance, multiple alleles, epistasis, and polygenic inheritance and pleiotropy. So let's get started with incomplete dominance. Okay, so incomplete dominance is something we see every day. So this actually is something that looks a lot more like the blending inheritance than well, so yeah, that's exactly it. It looks like the blending inheritance. So the idea behind incomplete dominance is that you have a genetic homozygous dominant phenotype. So in this case, it's RR. Oops, I've got a white pen. In this case, it's RR. Okay, so that's dominant for red. Then you have a recessive, which is little r, or something that will make white flowers. And these are snapdragons, for the example. So there are a number of different actual traits that can be accounted for by incomplete dominance, but in this case, the most well-known example is flower color and snapdragons. So you can think that, oh my gosh, what if Mendel had used snapdragons to do his research instead of using these lovely two-breeding pea plants? So when you breed a red flower with a white flower, in snapdragons, you end up getting an F1 that's pink. So the heterozygote is a pink flower. And in fact, what that means is that the red, or the big R allele, is not completely dominant over the white. So in this case, the example of snapdragons can be explained by the fact that red codes for a specific enzyme. So if enzyme 1 makes a protein, an enzyme that can make red, right? So you need a certain level of enzyme 1 to make red. Say you need, 100% enzyme 1 to actually make red color. Well, in this case, in this case of the heterozygote, you actually only have 50% of enzyme 1, right? So in this case, you can make enough red that you end up with a pink flower, but it turns out that, like with mixing paint, if you mix red paint and white paint together, what do you get? You get pink paint. Or if you put a red sock in your wash machine, or this red sweater that I was wearing today, if I throw that in with a bunch of white clothes, and it was the first wash, I might get pink clothing, because in fact, the red's not dominant, it's not going to make all my clothes red, but it's going to mix with the whites of my clothes, and it's going to make pink stuff. So in incomplete dominance, you get this phenotype that's essentially a dilution of the dominant phenotype. And in this case, it's because this enzyme, you need a certain amount to make red, and so if you only have part of that amount and the default color is white, then you end up making 2 pink flowers to every 1 red and 1 white. Okay? So incomplete dominance simply means that the phenotype is an intermediate phenotype. Again, this can be, this is very similar to blending inheritance, or the idea of blending inheritance. And in this case, in a monohybrid cross, if you have the F1s, so this RR here, so this is 2 heterozygotes in a monohybrid cross, you're going to end up with this 1 to 2 to 1 phenotype. Oops, let's see, 1 to 2 to 1, not 1 to 2 to 2, 1 to 2 to 1, where you get 1 red, 2 pink, and 1 white. So in this case, in an incomplete dominance, you get 1 dominant, 2 heterozygote or intermediate, and then 1 recessive. Okay, so then there's codominance. So the phenotype might look kind of similar to incomplete dominance. So with codominance, there's no such thing as a recessive. So in this case, both alleles are going to present in the phenotype. So in the example that I'm using, if you take a black hen and a white hen and you breed them together, you have two dominant WW alleles, two dominant BB alleles, and then you end up getting this black and white speckled hen. And that's because rather than, you're not diluting the black here like you would be with incomplete dominance. In fact, both alleles are just presenting, so you end up with a speckled black and white hen. So with codominance, both alleles are dominant, and so you get a split phenotype. So then there's the idea that there are multiple alleles to code for things. So with all of the examples we've been talking about so far, the idea is that there's only two alleles in the population. But we all know that, in fact, people don't just have brown eyes or blue eyes, right? There's not just big B and little b. And this is what I was talking about when I said that it gets more complicated than that, because we know that with everything like blood types, skin, hair, eye color, there's a range of phenotypes. With eye color, you can have dark brown, light brown, hazel, green, blue, red. Yes, albinos can have pink eyes. Mostly they have blue, but still, you can have all sorts of different options. Or hair, it goes from striking white to the darkest of black. And skin, same thing. You can go from super, super pale to very, very dark. But this is all accounted for by the fact that we've got a very big population, and our population has been evolving over time, right? So in Mendel's example, he was looking at peas, really simple peas that in fact had been inbred to start with. They were true breeding purple and true breeding white. And so therefore, those two colors of flowers, those were the only options. But with humans and other organisms, there are so many of us, and there's been so much evolution and adaptation and random mutation that we have more than just the two alleles in the population. So what do I mean by that? So anything that can be coded for by 3 or more alleles is a multiple allele phenotype. But you have to remember, even though there are more than 3 or more alleles coding for this, how many alleles does a progeny have? How many alleles do people have? We still only have 2 alleles, right? Only 2 in one individual. Right? So even though the population might have, 50 different alleles that code for certain things, you still only have 2 parents. And those 2 parents still only had those 2 chromosomes, the one chromosome that they each gave you. And that means that you only have 2 alleles coding for that. And that means that your progeny will only have the option of one of your 2 alleles. But in somebody else's family, they had maybe a different allele. So in terms of blood type, for example, you can say, okay, well, we know that we have A, B, and O in our population. So somebody could get 2 O's, one from each parent. They can get an A and a B from a parent, B and a B or a B and O, or A and O, right? So AO and BO really are just A and B type blood. But we've got all of these different blood types that we can get because there are 3 different alleles in our population for blood type. And then this is what it would look like. Basically, you have, so if this is the original allele or the ancestral allele, it has this gene sequence, okay? Over time, genes accumulate mutations, and that's what allows us to, that's another way that we can have variation. And so in this case, even though this is the original nucleotide here, maybe it's changed there, or maybe this nucleotide has changed, or maybe this one has changed. But the idea is there are 3 different changes which make these 3 different alleles, okay? So again, I was mentioning the ABO blood group. So this is the example of multiple different things, right? So the ABO blood group, there are 3 different alleles that code for these different blood types. It's the example of dominance and codominance. So we know that A and B are dominant to O, okay? So anybody with type A blood is going to be, and say they get, it's what I was mentioning, so say they get an A from one parent and an O from the other, they're going to be considered type A. So what are blood types? Blood types are basically you have a genotype, and your blood cells put on their surface glycoproteins. So if you remember glycoproteins, what are glycoproteins? They are transmembrane or integral proteins that have different carbohydrates on the end, and those carbohydrates help their body to know what are you. So they're like recognition proteins. And so somebody with a type A is going to have an antigen A, or basically the glycoprotein A on their blood cells, and their body's like, cool, we're type A. But when somebody puts a type B blood into their blood, their body's going to attack it, because it's like, oh, I'm pretty sure we're only supposed to have antigen A, but I just found this antigen B. What is that? And what ends up happening is if you get, say, a transfusion of the wrong blood type, then all of that other blood will be sequestered in your body, and it starts clumping, and it can kill you because your body is now attacking this blood. And that's why, for example, when somebody has a, what's the word? Oh my gosh, when one of their organs is transplant. So if you have an organ transplant, it's necessary that you want that organ to be as similar to yours as possible, because in terms of these alleles, our bodies know that, okay, so this person, like minimally blood type, but other, there are other things that we can make sure that they're similar. But if this person has an AB blood type and you have an AB blood type, at least your body won't attack the blood that was running through that organ, right? So it's really important to know that there are different alleles, and in fact with blood types you've got 3 different alleles, and A and B are dominants, and A and B are co-dominant, so that if you have an A allele and a B allele, you are type AB, period, because those can exist together if you have the alleles that are coding for them, but those are co-dominant, whereas O is recessive to both A and B. Okay, so this is the way that you write. So rather than writing the way we were writing it before, like, with a big P, little p, if you're looking at these different combinations of multiple alleles, you would write it more like a superscript rather than just the letter, right? So in this case, I is designating blood type, blood type. And so in this case, this is I to the A, so that would be an A allele, or I to the B, and that's a B allele, or little I would be O, okay? So when we're doing the Punnett square for this, you know that A and A make A, type A and B make type B, type AB, right? So this is type A, because it's just one antigen on the surface, it's A. This is AB because of codominance, so AB is codominance. And then when you mix little I or O with either A or B, you're going to get A or B by itself because of regular dominance. And then this little o, or not little o, little I is type O. It's the most recessive trait. These are these have actually no antigens on their surface. That's why O is the universal donor, because if you have blood cell A that's got, let's say it's got A antigens, blood cell B that's got B antigens, AB has AB antigens, and then O just doesn't have anything. So you can put O into all of these. Okay, so the next concept that changes Mendelian genetics is called epistasis, or epistatic mutations. So what this is when you have a change, or if you have any gene that is dependent on the presence of another gene, then it's called epistasis or epistatic interactions, okay? So if you have an epistatic mutation, it means that you've got a change in one gene, and that then changes another gene because of some relationship they have. So what this means is that you can have a dominant genotype. So say I have big B, but if I have this interesting epistatic mutation, then maybe my eyes are still blue. Okay, so I'm going to show you this with the Labrador example. So Labradors are really interesting because big B is dominant, black fur, right? So here you see big B is black fur. Heterozygotes make black as well, right? Because black is dominant. But if you have two little b's, then you get chocolate labs. So dominant is black, so B equals dominant. Little b equals recessive, and then the dominant is black, and the recessive is brown. But how do you get yellow labs? Because that's actually what I mostly see. Well, it turns out that there's this second gene, and that second gene either allows pigment or it blocks pigment. So epistasis means that even though the pigment color is determined by allele B, big B or little b, if you have big E, or if you have a specific gene E, then in this case, big E, then you allow pigment to form. So with big E equals allows pigment. But if you don't have E and you get these little recessive E's, then pigment is blocked and you end up getting yellow fur. So here, this is a very blurry version of a similar idea. So in all of these that have big E, pigment is allowed to form, right? So you get the black dogs, or big E with a little b, you get the chocolate labs because now pigment is allowed. But anything with the little e anywhere, oh, I guess it has to be homozygous for little e. My bad. Okay, so anyways, with a little e, a homozygous little e, then now pigment is blocked. So even though you have a big B coding for black or a little b coding for brown, the cells can't produce pigment, and so then you get a yellow lab. So then one of the other traits that can be confusing is polygenic inheritance. So I was using eye color as an example of big B, little b. But the thing is, a lot of these phenotypic traits that we have are created from multiple genes. So it actually turns out that eye color, skin color, etc., there's actually multiple genes that help to push your phenotype to a certain look, right? So in polygenic inheritance, there's multiple genes that work together to make certain proteins that work together to then make a phenotype. So they're called quantitative traits, which means that in quantitative traits, you have multiple loci, so multiple places on the chromosome that have to be expressed together to make a phenotype, right? So a quantitative trait equals multiple alleles expressed for certain trait, for a specific trait. Okay, so with polygenic inheritance, the traits are quantitative, and it's required that you have multiple genes that are expressed together at the same time to allow for this phenotype. And the way it works in a population is that usually you should get a bell-shaped curve for the phenotype. So height is a really good example of polygenic inheritance. It's not just that, oh, I have two tall parents. Well, if you have two tall parents, you're generally going to be tall. However, the way it works over the population is that it actually requires multiple different genes to make a tall individual. You have to have, something coding for long bones and something coding for growth and something coding for X and Y and Z. And in a population, you tend to get something that's similar to a bell curve shape, so that most of the individuals in the population are going to fall in the middle, right? So in average height. Oh my gosh. Oh my god. Seriously, spelling is an issue here. Average height. Okay. Anyways, but then you're going to have these outliers where they have all the genes coding for ginormous. So these are the basketball players, right? 6'3" and above. And these are I don't know what sport you play when you're super short, but these are the gymnasts, let's say. So you've got all of these different genes that are coding for a certain individual. Now, the reason you can say, well, if my parents are tall, I'm going to be tall, is because odds are if they're tall, they have all of those genes being expressed to make a tall individual. So when independent assortment happens, if they've got all the stuff that make tall people, and then you get them, you're going to be tall as well, right? Makes sense. But that's also why sometimes in a family you'll see that there's like a rando short person or a rando tall person, because maybe their mix just worked out perfectly to make that. The rest of their family is the other thing. Now, one thing to think about with polygenic inheritance is, well, it's true that height is determined based on multiple different genes being expressed at the same time together, but it can be modified by the environment. So if you take two twins and separate them at birth, okay, so they had all of the same nutrition and in the womb, and they're identical, so they have all the same genes expressed. But then you starve one twin and you give one twin all of the vitamins and nutrients that it needs, then this twin is going to realize its potential. And this twin won't. Sad face. So this is actually something really similar to the writing assignment that I gave you guys this week that's talking about economics. And so people have this genetic predisposition to intelligence, so if you have two really intelligent parents, sure, you're going to be intelligent too, but if you don't have the opportunity to realize that potential, then you're basically screwed. So you've got Shorty over here who had every opportunity to be tall genetically, but her environment didn't support that. So think about the difference and how the environment can actually affect genetics and polygenic inheritance. And then, so the final example is pleiotropy. So in pleiotropy, you've got one gene change, but it affects a lot of things in an organism. Okay, so we're going to go back to the sickle cell anemia example here. So sickle cell anemia, we know there's only one amino acid that's changed. So that could be as small as one or two changes in a gene, in a codon, right? So if you have three different, one codon change, so one or two nucleotides or whatever, is changed, that causes sickle cell anemia. However, it turns out that because your blood cells are doing this sickle shape versus the beautiful round shape that they're supposed to do, this can have physical effects that exhibit in sickle cell patients. So pleiotropy means that even though only one thing is changed, you get like system-wide effects. So in sickle cell anemia, if a homozygous recessive individual has sickle cell, right? What does that mean? It means that when they have abnormal hemoglobin, so when there's low oxygen, their cells take on this terrible sickle shape, okay? And when this happens, even though that's the only genetic thing that's changing and that's the only protein that's changing, you end up getting all of this terrible stuff that happens to you because your blood cells are abnormal. So you have lack of oxygen to the brain, your heart can fail, you're exhausted because your body's not getting the oxygen that it needs, you can have problems with your lungs, heart, kidneys, etc., all because this one amino acid has changed. Okay, so pleiotropy means that you might have one gene that's changed, but there are global or systemic effects. So that is the summation of the additions to sort of my Mendelian biology. And so please email me with questions or come to office hours because this will be on your quiz on Wednesday.